This video protocol demonstrates an effective technique to knock down a particular gene in an insect and conduct a novel bioassay to measure excretion rate.
The technique we are going to demonstrate today combines an RNAi-mediated gene knockdown with an in vivo diuresis assay. It can be used to study diuresis in different arthropod species, and we actually developed it to study the function of aquaporin water channels in 2 different mosquito species.
The protocol I will be demonstrating today is set up in 2 parts. The first demonstration illustrates how to construct a simple mosquito injection device and prepare and inject double-stranded RNA into the thorax of mosquitoes for RNA-mediated gene knockdown. The second demonstration illustrates how to measure excretion rates in mosquitoes using an in vivo bioassay. As the first step in order to perform RNAi-mediated gene knockdown, specific double-stranded RNAs against genes of interest and controlled double-stranded RNAs are synthesized. Additional equipment and materials that will be needed include an aspirator with collection vial for mosquito collection, fine-tip forceps, CO2 fly pad hooked up to a CO2 gas bottle, ethanol to clean the fly pad, stereo microscope with light source, needle puller if needed, glass capillary needles with marked 1 mm increments, a microinjector to inject mosquitoes, a container to place mosquitoes in after the injection, and a food source for the mosquitoes after injection, such as 20% sucrose-soaked cotton ball or water-soaked raisins.
A simple microinjector can be constructed by using scissors, a metal needle, a glass capillary, a 1 milliliter syringe.